For the first time, a new camera shows a high-definition view of how food travels through our bodies and into our stomachs. Food enters the stomach through a hole at the top. The stomach is a bag of muscle that churns, squashes, and squeezes food into liquid. At the same time, acids break the food down. The stomach walls protect themselves with a lining of mucus. Without it, the acids could digest parts of the stomach itself, causing stomach ulcers. About an hour later, the stomach squeezes the broken down food out through a tiny hole called the pyloric sphincter. The food enters the small intestine, an 11-foot coil of tube where we absorb most of the nutrients. The interior wall of the small intestine is lined with millions of microscopic projections called villi. These increase the surface area of the gut, making it easier to absorb nutrients. First, the pancreas pumps out a juice that neutralizes stomach acid. Then bile from the liver breaks down the fats into tiny droplets. Smaller droplets are easier for the intestine to absorb. After an hour and a half, the small intestine has absorbed most of the nutrients from the food. It's time for what remains to move on. It enters the large intestine through this, the ileocecal sphincter, a valve that keeps our food from going back where it came from. What's left is a mix of waste food and dead cells from the walls of the gut. The large intestine's main job is to extract water from it. Lots of bacteria live here too, but it isn't because of an infection. We actually need them. They produce enzymes that break down complex carbohydrates in our food, carbohydrates we couldn't otherwise digest. Finally, after about 12 hours, we expel what's left of our first meal.
Okay, so here we go at the digestive system. And with our digestive system, I like to tell the story of the tube and the squirters. So ultimately, we're going to have food enter our mouth and exit our anus. And along the way, we're going to take that food and we're going to break it down. And all of that, we cannot talk about this whole story without talking about enzymes. So enzymes are proteins. They typically end in ase, and they do not get changed in chemical reactions. However, biological reactions could not happen without enzymes. So if we take our substrate, like sucrose, which would be a disaccharide, a double sugar, it could, through the assistance of the enzyme, be broken down into monosaccharides or simple sugars like glucose and fructose. And you'll notice this water coming into this as well and getting involved in this process. So let's take a look at the major nutrients and the chemical reactions that take place. So if you see hexagons or pentagons, we're typically thinking sugars. So if we have a synthesis reaction, we are putting things together, we're building them up. And when we think of dehydration, we think of taking water out. So here we have two monosaccharides and we're putting them together into a disaccharide. As we put these together, this bond between them, water is also produced. Now, what we are talking about more so with the digestive system is not the buildup of or larger organic molecules, but a lot of times the digestion or the breakdown. And to lyse something is to split it apart. Hydrolysis, we've got some water involved here. So the hydrolysis of a sucrose, which is like table sugar here, you're going to have sucrase. Again, you need an enzyme to help make the biological chemical reaction take place, but water is also going to be involved here. So a complex molecule is broken down with the assistance of an enzyme, but with also the addition of a water molecule. So the water is going in here and assisting and is being part of the chemical reaction where these break apart. Now, dehydration synthesis and hydrolysis are not only involved in carbohydrate chemical reactions, they're also going to be involved with lipids. So you are going to have your dehydration synthesis, where you're putting together the glycerol and the fatty acid chains here. And but as you break them apart, again, you're going to have lipases, an ACE, an enzyme that's going to assist in the hydrolysis of triglycerides in your digestive tract. And that hydrolysis, water is going in to help split these apart in this direction, would be hydrolysis. Finally, with our proteins, they ultimately get broken down into amino acids. Average size protein is about 400 amino acids long. You hear peptide, you're also thinking protein. But again, all these organic molecules do follow this same process of dehydration synthesis, building up, and hydrolysis of breaking down. Again, we have to have enzymes which help break down our proteins, and we have different ones for the different sizes. But as we break them down, water is going to also come into the chemical reaction. The enzyme is going to help loosen up the bond, and we are going to break this dipeptide into the individual amino acids, which then ultimately we have to get them small enough to fit into our across the membranes of our digestive tract and into our bloodstream. Now, before we get any further, we should also mention the role of vitamins and minerals. So our body does not have the ability to build and make vitamins on its own. We have to consume vitamins. And the major function of vitamins are to function as coenzymes or cofactors. So with the other enzyme video, you have your protein enzyme. We have an active site where the substrate will bind, and ultimately the chemical reaction will take place and will create a product. But the active site in most enzymes is not completely the right shape until a vitamin binds into the active site, and now the substrate can actually fit. So that's the role of most vitamins, is to act as coenzymes or cofactors to help with chemical reactions. So we have this giant chart of digestive enzymes, where they're from and their functions. And again, a lot of these, the names kind of tie into what they're breaking down. Amylase breaks down amylose. Lipase breaks down lipids. But we can just memorize a chart, but I always find it easier to kind of associate with pictures and try and tell a story. However, before we get there, I'm going to talk a little bit more about enzymes because a lot of people think it's, without really getting into digestive enzymes, they're like, oh, it's the stomach acid that breaks down your food. And in reality, that's not the case. We still have this idea of lock and key model, and notice this has an O-gin at the end. So this pepsinogen is what we would call a zymogen. Again, it's a non-functional form of the enzyme because it's the wrong shape. And I'll come back to this and explain it in a little bit more detail. But we should talk a little bit about pH, because as we start talking about gastric acid or stomach acid, it has a pH of 1. And our pH scale, as a reminder, 7 is neutral. We get up to 14, that's very alkaline or basic. And we get down towards 1, and we have a very strong acid. Now, where do these numbers come from? It's about the hydrogen ion concentration. So most things that have a pH of 7 are going to be the same amount of hydrogen ions and hydroxide ions in concentration. They'll be both 1 times 10 to the negative 7th molar. So here, if something is an acid, what that means is it has a much larger amount of hydrogen ions. And an alkaline or a base has a very small amount of hydrogen ions. So one way to picture this is if it has a low pH, it's got lots of hydrogen ions. If it has a high pH, it has a lot of hydroxide ions and very few hydrogen ions. Now, hydrogen ions, they're an oxidizing agent. What that means is if you go back to some of your chemistry, you've got the oil rig. Oxidation is losing. So if it's an oxidizing agent, it's going to help take electrons away from other things, which is going to like speed up chemical reactions and can be very reactive, right? Sometimes that's good, sometimes that's bad. So different areas of your body have different pH. Enzymes don't all work at the exact same pH. Some work best at different pHs. For example, the gastric acid, as you look here, has a pH of 1. That's hydrochloric acid. It's meant to kill bacteria that is on your food. It helps liquefy the food, but it also activates pepsinogen to become pepsin to break down the protein. So the protein digestion begins in your stomach with pepsin. So, however, as this hydrochloric acid and the mixture from your stomach starts traveling down your digestive tract, you don't want to burn up all of the other areas that aren't lined in as thick a mucus. So the pancreas is also going to secrete digestive enzymes, but also bicarbonate into the duodenum, which is the beginning of your small intestine, to kind of buffer out and kind of neutralize a little bit of this pH. And ultimately, the pancreas secretions have a pH of 8.1. So, tubes and squirters, right? In through your mouth, down through your esophagus, to your stomach, to your small intestine, to your large intestine, and then out your anus. So, as we're traveling here, we're going to talk about your stomach with this pepsinogen. So again, the digestion of carbohydrates begins in your mouth, but then that gets turned off and the protein digestion begins in your stomach. And it's this part of your duodenum and the beginning of your small intestine where the vast majority of all of the digestion is taking place, and ultimately has to get things small enough so that it can be absorbed by your small intestine. But let's focus on your stomach a little bit here. So the stomach lining has all these, look at, oh, a gland, a gastric gland secreting onto the surface. So this isn't an endocrine gland, which would secrete into your blood. It's secreting onto the surface of the stomach lining. So what is getting secreted? Well, mucus, but also pepsinogen, which will become pepsin, and also hydrochloric acid. So all of these things are being secreted into your stomach. So as we are shooting this out, pepsinogen is what we would classify as a zymogen. The hydrochloric acid in the reaction with the hydrochloric acid ultimately turns it into an active enzyme because part of the peptide is removed. This enzyme is now the right shape to break down dietary proteins. So imagine this is the lining of your stomach. These are the cells alongside your stomach. This is in the tube, okay? Inside the stomach, it's in the tube from your mouth to your anus, right? And it's heading to the small intestine so the small digested nutrients can diffuse into your blood. Now you're eating proteins, but you're also having proteins obviously inside your cell, right? And these are important proteins for cell function. So do we want an active enzyme breaking down the important proteins all over inside of our cells? The answer is no. So when pepsinogen is created, it's created in a form that is inactive, and it's only when it's in the right environment that it will become active, and then we can get into that lock and key model. It's the right shape. Now again, the lining of our stomach has all this mucus on it, which would protect it from all these hydrogen ions from the hydrochloric acid, right? So the cells lining your stomach are going to secrete pepsinogen and hydrochloric acid into the tube of your stomach, right? The hydrochloric acid, the hydrogen ions are going to interact with the pepsinogen and turn it into pepsin. So right here we got pepsin, and now it's the right shape to actually interact with the proteins from your food and break them down into smaller peptides so that they can ultimately be broken down into amino acids. Okay, so the story. Again, this looks kind of crazy because it's so long. However, if we take it a little bit at a time, you can eat an elephant one bite at a time, but you can't eat an elephant in one bite. So, there's my digestion story joke, or advice, I guess. So, again, from our mouth to our stomach, duodenum, jejunum, ileum, cecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, anus. And then, so that's our tube, right? And the inside of the tube is oftentimes still considered the outside of our body. And it's kind of weird to think about it that way, but that's one way, well, a good way to think about it. We also have squirters like our salivary glands, our pancreas, our gallbladder, and the tube itself is going to squirt in some enzymes as we go. So let's start at the mouth. Once upon a time, food enters your mouth. We've got, and again, this is kind of the tube right here down this list. And then you've got some squirters in here. What are they squirting? Which enzymes are involved? And what those enzymes actually do to the food. So, again, food enters your mouth. Salivary glands secrete amylase to break down complex carbs. Those big amylose is like starch. The bolus, which is what you've chewed up, goes through your pharynx, the back of your throat, down the esophagus, into the stomach. Pepsin is secreted by the stomach to begin breaking down proteins. The hydrochloric acid also shuts off the amylase, so that enzyme that was breaking down the carbs is now shut down. So carbohydrate digestion stops in your stomach. The mixture in your stomach is called chyme now and enters the duodenum. Okay, so again, lots and lots of stuff is happening in this first little section of your small intestine. So bile is made in your liver, and it's stored and secreted from your gallbladder into the duodenum. Now, what bile does is it isolates those fat molecules. It gets each triglyceride kind of by itself. Now, let's stay with that fat in here. Okay, so the bile isolates the fats. The pancreas is going to secrete lipase. It's an enzyme, right? And that's going to break down those triglycerides. It's going to break down the fats. The amylase got shut down in the stomach, right? So the carbohydrates are not fully digested. So the pancreas is going to send out amylase for the carbs, and trypsin. Again, we had pepsin here. Now we're secreting trypsin for the polypeptides. Again, that's the smaller version of the proteins. The pepsin got them down to polypeptides, and then the trypsin is going to get them a little smaller. So the duodenum also is going to secrete maltase, sucrase, and lactase to break down those disaccharides and get them down into monosaccharides and get them down to like glucose size. Finally, it's also going to secrete peptidases. Again, here's our ACE, it's an enzyme, and it's going to break down those peptides. So we've got pepsin, trypsin, and then peptidases, which ultimately gets the peptides down into amino acids. So from the duodenum, the simplest forms of nutrients can be absorbed by the jejunum and the ileum into the bloodstream to be directed by the hepatic portal system to the liver and then ultimately to the heart and the rest of the body. I'll make another video for that. Meanwhile, the remains of the chyme, the undigested and unabsorbed material, continue from the small intestine to the large intestine, where most of the water is absorbed. So we go here from the cecum, which is the first part. We'll go up the ascending colon, across the transverse colon, down the descending colon, across the sigmoid colon. Now, when you eat food, especially, you hear about fiber, and fiber is that tough fibrous protein or not protein, the tough fibrous carbohydrate that you can't actually digest, and it's good for your guts, right? It's good for your digestive tract. It's good for your colon. Well, that fiber actually feeds the bacteria, and those bacteria end up generating vitamins because, again, we don't make vitamins on our own. We have to get vitamins from the bacteria or from our food, and those vitamins can act as coenzymes, even in like blood clotting and stuff. So, again, ultimately, the remaining waste is stored in the rectum and eliminated out the anus. So that's our story here. Again, you learn your tube, learn your squirters, and learn what they are squirting. So through repetition, you should be able to take a diagram like this and walk through it and tell me, name the parts of the tube, what's squirting in enzymes into the different parts of the tube, and what is being broken down at different stages and what's being absorbed in different stages. So that's the digestive system. I'll give you a couple other ways to visualize this, which might also be helpful.
Digestion starts in the oral cavity, where the food is moistened with saliva and chewed. Food bolus is formed to facilitate swallowing. Saliva is secreted by the salivary glands and contains the enzyme amylase. Amylase breaks down starch into maltose and dextrin, which are processed further in the small intestine. The food bolus is propelled down the esophagus into the stomach, the major organ of the GI tract. The stomach produces gastric juice containing pepsin, a protease, and hydrochloric acid, which act to digest proteins. At the same time, mechanical churning is performed by muscular contraction of the stomach wall. The result is the formation of chyme, a semi-liquid mass of partially digested food. Chyme is stored in the stomach and is slowly released into the first part of the small intestine, the duodenum. The duodenum receives the following digestive enzymes from accessory organs. Bile, produced in the liver and stored in the gallbladder. Bile emulsifies fats and makes it easier for lipases to break them down. Pancreatic juice from the pancreas. This mixture contains proteases, lipases, and amylase and plays a major role in digestion of proteins and fats. The small intestine also produces its own enzymes, peptidases, sucrase, lactase, and maltase. Intestinal enzymes contribute mainly to the hydrolysis of polysaccharides. The small intestine is where most of digestion and absorption take place. The walls of the small intestine absorb the digested nutrients into the bloodstream, which in turn delivers them to the rest of the body. In the small intestine, the chyme moves more slowly, allowing time for thorough digestion and absorption. This is made possible by segmentation contractions of the circular muscles in the intestinal walls. Segmentation contractions move chyme in both directions. This allows a better mixing with digestive juices and a longer contact time with the intestinal walls. The large intestine converts digested leftover into feces. It absorbs water and any remaining nutrients. The bacteria of the colon, known as gut flora, can break down substances in the chyme that are not digestible by the human digestive system. Bacterial fermentation produces various vitamins that are absorbed through the walls of the colon. The semi-solid fecal matter is then stored in the rectum until it can be pushed out from the body during a bowel movement.